Hey everyone, my name is Raoud and I'm one of the PGY3 general surgery residents at UAB in Birmingham, Alabama, and today I'm going to talk about a part of my research in Dr. Elizabeth Byerly's lab. I have no disclosures. Patients presenting with metastatic hepatoblastoma have limited treatment options and survival rates are as low as 25%. We've previously shown that PIM3 kinase not only promote tumorigenesis, but also tumor metastases in hepatoblastoma by promoting migration, invasion, and anchorage-independent growth in vitro and formation of lung metastases in vivo. We've shown that overexpression of PIM3 kinase led to increased metastatic burden in a tail vein injection model. When hepatoblastoma cells, either overexpressing PIM3 or transfected with an empty vector control, were injected in tail veins of thymic nude mice, the mice were monitored for in vivo bioluminescence to detect lung metastases, and their lung harvested, sectioned, and stained with H&E. We found that PIM3 overexpression resulted in an increased metastatic burden compared to control hepatoblastoma cells. We've also shown that PIM3 overexpressing cells upregulated cell surface expression of CXCR4 when evaluated by flow cytometry. This is a bar graph quantifying percent CXCR4 positive cells showing that it's significantly higher in the PIM3 overexpressing cells compared to the empty vector control hepatoblastoma cells. So what is CXCR4? It's a chemokine receptor, basically a cell surface receptor, that's been shown to be a key regulator of cancer metastases and has been implicated in migration and invasion, as well as chemotaxis of CXCR4 expressing cells towards secondary organs for metastases. So we asked the question, does PIM3 act through CXCR4 to promote hepatoblastoma metastases in vivo? We sought to determine whether CXCR4 contributes to the PIM3-induced formation of lung metastases in vivo, and whether blocking CXCR4 with a small molecule inhibitor, AMD3100 or plerixafor, would decrease the metastatic burden of PIM3 overexpressing cells. To assess the biological significance of PIM3 activation of CXCR4 signaling, we took RNA sequencing data of hepatoblastoma patient tumors, 34 samples, that we obtained from one of the few publicly available hepatoblastoma data sets. We used the Spearman rank correlation coefficient analysis, and we evaluated the correlation between gene expression of PIM3 and CXCR4. And we found that PIM3 gene expression correlated positively with CXCR4 gene expression in hepatoblastoma tissues, supporting the notion that PIM3 may regulate expression of CXCR4 in hepatoblastoma. So onto our in vivo experimental design, we injected PIM3 overexpressing cells, HUH6 human hepatoblastoma cells, in the tail vein of thymic nude mice who were then randomized to receive either subcutaneous injection of the CXCR4 receptor antagonist, AMD3100, or a vehicle control, in this case PBS. And we treated these mice for 5 days a week for 6 weeks. At the study endpoint, we harvested the mice's lungs, sectioned them at 3 different levels from each animal, stained them with H&E, and imaged them using light microscopy. And we quantified the metastatic burden and found that mice treated with AMD3100 had significantly decreased pulmonary metastatic burden of PIM3 overexpressing hepatoblastoma cells compared to mice treated with vehicle. This is representative H&E images from both groups with dotted lines highlighting the metastatic lesions. In conclusion, we've shown that treatment with AMD3100 reduced pulmonary metastatic burden in animals injected with PIM3 overexpressing hepatoblastoma cells, corroborating CXCR4 as a plausible mechanistic link between PIM3 and its promotion of hepatoblastoma metastases. I would like to thank my mentor, Dr. Elizabeth Byerly, and members of the Byerly Lab, and our funding resources. And with this, I would like to take any questions. My contact info is on the top right corner of the slide.
Thank you, Raoud. This was an interesting, elegant, straightforward experiment that non-basic science people can understand and see clinical applicability for. And I just wanted to ask you the time point where you introduced the antagonist during the experiment.
Is there a way to go back, I guess?
Yeah.
Oh, sorry. This one? So
there.
at the mice were injected, I was trying to show the design. So mice were injected with the cells, and then at day 0 is when they were randomized right away to receive either the vehicle control or the AMD 3100. So we were basically looking at, we've already done research showing that PIM3 hepatoblastoma cells compared to control cells increase the metastatic burden. So basically we're evaluating the ability of cells to extravasate and form metastatic lesions.
So
With blocking CXCR4.
It was right away.
Yes.
And then I wanted to ask you about it is hepatoblastoma cells. Are there other cell lines or pediatric tumors or other tumors that may involve this similar type of PIM3 mechanism?
So our lab have looked at PIM3 kinase and its role in other pediatric cancers, such as neuroblastoma. There are not many. This is actually, HUH6 is one of the only commercially available cell lines that you can buy for hepatoblastoma, so it kind of limits the research. We've also used PDXs, patient-derived xenografts, to look at PIM3 and its role in hepatoblastoma.
Interesting. So if you had a dream of clinical applicability for this, you would think of what?
So we're advocating for PIM3 being a target in hepatoblastoma. I feel like we've done in our lab a collective amount of research showing that PIM3 is important not only in tumorigenesis but metastases. So we're PIM3 inhibitors right now, such as like AZD1208, is in clinical trials for other tumors but not pediatric cancers. So I know I kind of went into the mechanism of why we think PIM3 and how it promotes metastases, but I think it offers a really good target for therapy in hepatoblastoma for kids.
Fascinating.
So along the same lines, do you know if there are other cell lines that have CXCR4 as a means of metastatic escape?
Yes, so there has been research done in prostate cancer showing that CXCR4 similarly is important for chemotaxis of the tumor cells that are overexpressing CXCR4 into lungs or other secondary organs. So that's kind of where the research is going. And then there are AMD 3100, the drug that we used here, is in trials for not necessarily inhibiting metastases, but chemotaxis of cells and ability to recruit cells into the bloodstream.
Great. Thank you very much.
Thank you.